Good morning, everyone. Today, in this lecture talk, we are understanding chain termination or Sanger's dideoxy method of DNA sequence. Okay, before I begin, let us go through today's quality phrase. You may not be able to do all you find out, but you should find out all you can do. Okay. Become a good reader, become a good listener. Okay, all successful people around the world are good leader. Just read, become a good reader. Meanwhile, listen to other lectures, listen to me, and make up your own mind. Don't be a follower, be just a student. Be a good reader, be a good listener. Okay? When you become a good reader and a good listener, you will be able to enhance your reading and listening skills. And these skills which helps to improve your quality of life. Thank you. Now let us go through today's class DNA sequence by Sanger's method. Okay. So what is this DNA sequence? DNA sequence is nothing but determination of nucleotides or base sequence of DNA molecule or fragment. Okay. This is the most important technique available to the molecular biology. Okay. Here, what are we doing here? We are doing the determination of the precise sequence or order of nucleotides in a sample of DNA molecule. Okay. Our present ability to sequence essentially any DNA molecule is the result of four major developments. Okay. They are discovery of restriction enzymes, gene cloning techniques, gel electrophoresis methodology, and two different approaches of rapid sequencing of DNA molecule. Okay. Here in this case, the most important was discovery of restriction enzymes and their use in preparing homologous samples of specific segments of DNA molecule. Just imagine, without restriction enzymes, we cannot think about any genetic engineering experiment or any biotechnology experiment. Everywhere we need this particular one of the major tool, that is restriction enzymes, which we are using this to cleave the DNA molecule, where we are making larger DNA fragment into smaller DNA fragments. Okay, a second major development of gene cloning techniques, that is greatly facilitate the preparation of large quantities of a particular gene or DNA molecule. Even through this technique, we are able to construct many vectors, right? And we can also screen the desired DNA fragment, and we can get n number of amount of gene. Okay, that is desired gene we are able to obtain through gene cloning techniques. Okay. Next one is a third major advance was the development of gel electrophoresis procedures, where the DNA fragments that differ in length by a single nucleotide could be resolved. Okay, that is very, very important technique, gel electrophoresis technique. We have SDS page, right? Next comes finally two different approaches to the rapid sequencing of DNA molecules were developed in the period from 1970 to 1977. Okay, so these are the four major developments which were happened in the world of molecular biology. So what are those two different approaches? Chain termination or Sanger's dideoxy DNA sequence. This is the one of the DNA sequence method. Other one, chemical degradation or Maxima Gilbert method. Here I've given the definition. Here two different approaches of DNA sequencing. One is chain termination method. Other one is the chemical degradation method. Okay. So you have in your syllabus, you're studying only the Sanger's dideoxy method of DNA sequence. Okay. For understanding the function of a gene, it is essential to do characterization of DNA. Okay, this can be done by knowing the nucleotide sequence. Okay. You may ask question here why we need to do all this DNA sequencing. That time you can take this particular answer. To know the functions of DNA and to characterize one has to study the alignment of the nucleotides. Okay, what do you mean by DNA sequence? DNA sequencing is nothing but determination of nucleotides or base sequence. It means we are understanding the order of the nucleotides in a DNA molecule. Why we need to understand this? To know the functions of DNA, right? And to characterize, one has to study the arrangement of the nucleotides. Once we understood the arrangement of the nucleotides, next what happens? We'll be able to think about the um, amino acids. They're able to think about the genetic codons. Then amino acids, we can also think about what kind of proteins we are going to get, right? So this will give you the answers. Or you can also ask again question that why DNA sequence? Scientists need to know the sequence of bases because it tells them the kind of genetic information that is carried in a particular segment of DNA. For example, they can use the sequenced information to determine which stretches or fragment of DNA contain which particular gene, right? For example, insulin gene we discovered, so we are able to go for the production of the insulin, as well as to analyze the genes for changes in sequence called mutations that may cause disease. Okay, the DNA sequence which were mutated, the DNA sequence which are not undergone mutation, once we study their sequence characteristic, then we can decide about their characters, right? So that's why you need this particular DNA sequence. And the analysis of DNA sequence will also reveal the amino acid sequence, then molecular weights of proteins, gene regulation, and molecular cloning studies will be very useful when we perform this DNA sequencing. Okay, now let us go through here two different techniques of DNA sequence. That is, both DNA sequencing were developed simultaneously. Okay, first one is chain termination or Sanger's dideoxy method DNA sequence. Okay, which was given by Dr. Frederick Sanger and A.R. Coulson in UK during the year 1977. Okay, whatever he explained this particular DNA sequencing methodology, still we are using this particular DNA sequencing. Okay, the next DNA sequencing was chemical degradation or A. Maxima and W. Gilbert method of DNA sequence, which was demonstrated and designed at Harvard University, USA. Okay, so we have here two different techniques but equally valuable, which allows the DNA sequencing of several kilo bases and can be determined in the minimum of time. Okay, so you must keep in your mind what are two different DNA sequencing method. If you were asked by the examiner during your viva, you must be able to give this particular answer. Okay. Now let us go through some more details about our Frederick Sanger sequencing. Okay, Sanger and co-workers in the year 1977 invented a new method for DNA sequencing through enzymatic polymerization. When I'm using here the enzymatic polymerization, you must keep in your mind by using this can also be known as enzymatic DNA sequencing. Okay, I'll repeat. Sangers and co-workers during the year 1977 at University of Cambridge, they developed the new method for DNA sequencing through enzymatic polymerization that basically revolutionized the DNA sequence technology. Okay, and this is the most important popular method of sequencing called dideoxy method, okay, or Sanger's method, which is named after its inventor, okay, Frederick Sanger, who was awarded Nobel Prize in Chemistry for his achievement. Okay, this particular DNA sequencing involves in vitro DNA synthesis. It means in a laboratory by using the glass vials or test tubes in the reaction tubes we are doing this particular sequencing methodology. Okay, based on the principle and biochemistry of DNA application, this DNA sequencing was done. So what is happening in the DNA sequencing? In DNA sequencing, what is happening here? That this is the technique by which the precise order of nucleotides in a DNA segment can be determined. Okay, we have a genome. Genome, that is nothing but the collection of genes. We cannot perform together this experiment by taking the genome. So we need to select a gene, right? So once you select a gene here, within the gene, what is the arrangement of the nitrogenous bases? This is the arrangement of the nitrogenous bases. How you got this one? This you got through DNA sequencing. This arrangement, the beads of nitrogenous bases A, T, G, C, A, C, T, T, G, A, T, C. This arrangement of the nucleotides we have understood through DNA sequencing. So we can say here the technique by which the precise order of nucleotides in a DNA segment can be determined by DNA sequencing. Okay, that is nothing but DNA sequencing. We are understanding the arrangement of the nitrogenous bases. Once we understand the arrangement of nitrogenous bases, you can think about what kind of mRNA this DNA will give. Okay, after the mRNA, from this mRNA, we can also think about the amino acid sequence. With the collection of the amino acid sequence, you can think about the proteins, which protein this gene which is going to express. Okay, in this particular in vitro method of DNA sequencing, we are using here the template. The most important thing is that during the polymerization process, DNA polymerase always need a template, right? So this is the template, DNA template stand we need in this particular experiment, right? And then we need a primer. Okay, this is primer we need. Other than primer, we need DNA polymerase. Okay, and we need DNA template strand, right? Then we need here the deoxynitrogen triphosphate. Individual nitrogenous bases you need here. Okay, once all requirements you have, then DNA synthesis will begin, as you've understood that in the in vivo DNA synthesis or DNA replication, right? Once the primer is ready in the in vitro DNA synthesis, what happens even in the in vivo, the DNA polymerase goes and attached to this particular region, right? To the primers. In this case, what happens? DNA polymerase enzyme cannot initiate by themselves. They need themselves on the template DNA. They need compulsory the primers. Okay. So what happens here? Once a gene or DNA fragment has been cloned, it is further subjected for the DNA sequencing. Okay, during DNA sequencing, we need compulsory primer. What is this primer? This primer is a single stranded known fragment. Okay, the first step in chain termination sequencing experiment is to anneal short oligonucleotide. Okay, this is the short oligonucleotide primer to the template. Okay, this primer which acts as a starting point for the complementary strand synthesis reaction which is carried out by the clinofragment of DNA polymerase. Okay, the primer is needed because template dependent DNA polymerase are unable to initiate DNA synthesis on an entirely single-stranded molecule. Hence, a short double-stranded region is needed to provide a 3' end onto which the DNA polymerase go and bind and can add new nucleotides. Okay, primers are having the free 3' end for the polymerization purpose. Okay, hope you understand here. Even in the DNA replication also they need compulsory the primer. Okay, to that primer, once primers goes and binds here, then DNA polymerase go and attach to the double stranded initiation region, then start synthesizing the individual complementary nucleotides. nucleotides. Okay. So you can see here as and when the DNA polymerase moved from here to here, they started already adding the complementary nitrogenous bases according to the template DNA. What this happens in the in vivo DNA synthesis. Same we are going to follow in the in vitro DNA synthesis. Okay. So here in DNA sequencing or DNA application, what is important here? Primer is very essential for DNA synthesis, right? The prime without primer, DNA synthesis is not possible. And always this primer provides initial 3' hydroxyl region. Okay, this is the 3' hydroxyl region group to form phosphodiester bond with the incoming DNTPs. Okay, so between the one nucleotide and the nucleotide, we can see the bond, that is the phosphodiester bond. Adjacent nucleotides which are connected through the phosphodiester bond, but opposite nucleotides which are connected through hydrogen bonds. Okay. So here you could see the formation of phosphodiester bond as and when DNA polymerase adds individual nucleotide according to the template DNA. There what happens? Phosphodiester bond will be forming between the adjacent nucleotides, and also meanwhile hydrogen bonds also will be formed during the DNA synthesis. Okay, hope you're following. Now, let us understand here who is this Frederick Sanger. Okay, Frederick Sanger, who got the Nobel Prize for explaining the structure of insulin in the year 1958. Okay, and also about the sequence of RNA virus during the year 1980. Look, let me go through some more details about Sanger's method of DNA sequencing. Okay, so one of the more major method for sequencing a DNA fragment is the Sanger method. Frederick Sanger, who won the Nobel Prizes for explaining the structure of the insulin in the year 1958 and the sequence of bases of an RNA viruses during the year 1980. Okay, the Sanger method is also known as chain termination. Okay, why chain termination? Because for how it works and due to the dideoxy method for the biochemicals it was. Okay, so this Sanger's method of DNA sequencing can also be known as chain termination. Why chain termination? Because we are using in this Sanger's method a modified nucleotides DDNTP. Just make a note. I'll make you understand in the next slides what is this modified nucleotides DDNTP. Okay, usually you'll ask this kind of question during Viva, even in the entrance examination. So this DNA sequencing can also be known as Sanger's method or chain termination. Okay, once these modified nucleotide DDNTP were added, their chain termination will happen or cleavage will happen. Okay, just they'll stop, further no more synthesis. So it was called chain termination. This can also be known as what method? Dideoxy method of DNA sequencing. Okay, keep this in your mind. The chain termination method requires a single stranded DNA. Okay, to perform the determination of DNA fragment. So the molecule to be sequenced is usually cloned into an M13 vector. Okay, M13 vector, you know, right, is one of the bacteriophage. If you want single stranded DNA molecule, if you could introduce double stranded DNA molecule to M13 vector, from the M13 vector you are going to get the single stranded DNA. Okay, so you can also think about that. To perform this DNA sequencing, we need compulsory single stranded DNA. If we are providing the double stranded DNA, the double stranded DNA will be subjected to the heat denaturation. Okay, and after heat denaturation, you are going to get the single stranded DNA, or else you can also obtain through M13 vector. Okay, or it may be denatured by heating. Okay, this is because chain termination sequencing involves enzymatic synthesis. Okay, during enzymatic synthesis of a second strand of DNA which are complemented to the existing template. Okay, they need compulsory template DNA. All DNA polymerase synthesize on the template DNA only. So we need to provide the single stranded DNA fragment for the polymerization process. This technique was the DDNTP in DNA synthesis has the name Sanger's dideoxy, DD, dideoxy nitrogen triphosphate is known also known as Sanger's method or chain termination method. Okay, in this DNA sequencing, it is very important to achieve both specificity and efficiency. What is specificity here? The primer anneals um only to the specific desired DNA portion, okay, and that should provide the initiation of double standard region. Then coming to the efficiency, referring to the number of template molecules that anneal to the primer, okay. So in this DNA sequencing, both specificity and efficiency of the primers are very important. Primers should go and bind properly to the template DNA, and they must provide 3' hydroxyl region, okay. This primer binding is very important. If with the proper specificity and efficiency, this primer should go and anneal or bind. If there is no proper specificity of this primer, then they will not able to give this 3' initiation region, and DNA polymerase cannot bind over there, okay. So the specificity and efficiency of the primers are very important during the DNA sequencing, okay. So in DNA sequencing today I'll be discussing with you principle, procedure. In this procedure, when you select any DNA molecule, if it is a double stranded DNA, you can subject for the heat denaturation, where double stranded DNA will get converted into single stranded DNA. Once the DNA is single stranded, then primers which goes and binds are anneals, which is of length of around 20 base pair. These primers are known single stranded fragments. Okay, then after primer annealing, what happens? Synthesis will begin in the presence of DNA polymerase, which goes and binds to the double stranded initiation region along with the primer. Then whenever this DNA polymerase adds this DDATP or modified nucleotides, their chain termination will happen. Okay, after this process, this particular reactions will be subjected for the denaturation through the electrophoresis methodology. Then the electrophorused DNA fragments will be subjected to the autoradiography. Then sequence termination we are going to find out. And next in slide you will also make about the limitations. So all this I'm going to cover in today's slide related with DNA sequencing. Okay. Now let us understand first and foremost important thing, the principle behind this DNA sequence. Okay, in DNA sequencing, we are using, please make a note, we are using here DDNTP. DDNTP, that is nothing but, make a note, dideoxy nitrogen triphosphate. Okay, if you want to do any in vitro replication or in vivo replication, you need compulsory a 5' phosphate and 3' hydroxyl DNA molecule, right? Compulsory, we need the DNA molecule which ends with the single stranded DNA, with one is the 5' and another one is the 3' hydroxyl strand, okay? And we need primers also, right? Primers, and we need the template DNA molecule, right? We need the template DNA molecule we need, and we need DNTPs, and we also need here the DDNTPs. Okay, so in the first structure, here you are able to make out here, this is the NTP. Okay, NTP means that is nitrogen triphosphate, and this is specifically ribonucleotide. Okay, when you take any nucleotide, you'll be having the base and you'll be having the sugar, and this will be the phosphate. Okay, this is the first carbon atom, second carbon atom, third, fourth, and fifth carbon atom. So when we have the hydroxyl group at second prime and the 3', right, we'll say it's a ribonucleotide, right? Whereas let us take the next nucleotide, that is deoxyribonucleotide. In short, we are going to say this as DNTP, okay? So in this case, DATP, that is nothing but deoxy nitrogen triphosphate, or we can say deoxyribonucleotide. What is this deoxyribonucleotide? So first carbon atom second carbon atom. At the second carbon atom, you don't have a hydroxyl group here, okay? Second primary atom, here hydroxyl group here. As in this case, in DNA molecule, there is no hydro oxygen group at the second carbon atom. So we mentioned here 2' deoxyribonucleotide, or in short, just we'll say deoxyribonucleotide triphosphate, right? So you can make out here, this is the triphosphate, 3 phosphate molecules are there. So deoxy, there is no oxygen group, oxygen groups were removed. So it's a deoxy. We can see that in the DNA. So this you can see in the RNA molecule, and this DNTP you can see in the DNA molecule. Okay, and this is the most important one what we are using in the Sanger's method of DNA sequencing, this which brings the chain termination. Okay, so what is this? It's a dideoxyribonucleotide. Keep in your mind, DDNTP means di means 2, deoxy means mole of oxygen, dideoxy nitrogen triphosphate. So in this case, what has happened in DNA molecule, as usually at the second prime carbon atom, no oxygen group, so deoxy. Whereas we mentioned here di means 2. At the 3 prime group also, in DNA sequencing, they have removed the oxygen group from here also. Okay, so this is the modified nucleotide. Okay, so this modified, by using this modified DDNTP, we are doing this particular Sanger's method of DNA sequencing. So this method can also be known as dideoxy method of DNA sequencing. So what is the full form of DDNTP? Dideoxy nitrogen triphosphate. So this is the principle behind this particular DNA sequencing method. Okay, here we can make it. Okay, this is the DNA strand and this is the primer, 5 prime phosphate. Okay, and this is the 3 prime hydroxyl group. We need a template. Okay, other than this normal DNTPs, okay, we need these modified DDNTP and modified DDCTP. Okay, we know better for the synthesis of the any kind of DNA molecule, we need one end must be compulsory, 3 prime hydroxyl and 5 prime phosphate molecule. Okay, in this particular modified DNTPs, okay, when at the 3 prime region, when there is no oxygen group, when there is no hydroxyl group, what happens further? No more synthesis. Okay, because when you take any kind of nitrogen DNA fragment, one end will be 5 prime phosphate group, other end will be 3 prime hydroxyl group, right? This is 5 prime phosphate and this is the 3 prime hydroxyl group. For the joining of the incoming nucleotide, if this is the 3 prime, the other nucleotide will be 5 prime phosphate and the 3 prime hydroxyl group. Okay, if I want to join here, they need compulsory 3 prime hydroxyl group. When I have removed the oxygen group from this region and there is no hydroxyl group, what happens when DNA polymerase adds this kind of modified nucleotide? What happens? There won't be any kind of further chain synthesis and reaction will be stopped in that particular region. Okay. So here you can make out the difference between the DNTPs and DDNTPs. So this is dideoxynucleotide phosphate. Okay. So you can make out here DDNTPs don't have this oxygen group at 3 prime hydroxyl. You know better, 3 prime hydroxyl very important for the polymerization of process. Whereas this is the normal NTP, that is DNTP, that is deoxy. Only one oxygen group which is removed from the second prime carbon atom, and that is the DNTPs. So the same thing I've written here. Sanger DNA sequencing techniques makes use of modified deoxynucleotides. So it is known as dideoxy nucleotides DNA sequencing. What has happened here? This is a deoxynucleotide. 3 prime hydroxyl will be there. Whereas in dideoxynucleotide triphosphate, hydroxyl molecule will be there. Instead, only the hydrogen will be there. That is the main difference in that particular presence and absence of the uh, uh, hydrogen molecule or hydroxyl in that particular DNA molecule. Okay. If we are going to say any DNA molecule, while performing this DNA molecule, we need compulsory DNA molecules. In that, we need nucleotides. Okay. So what is this nucleotide? When are you going to say this that as a nucleotide? When you are going to say nucleotide, when you have this phosphate molecule, when you have this sugar, and when you have the particular nitrogenous bases, you can say this as the nucleotide. Okay. Whereas in this particular nucleoside, when you are going to say nucleoside, when we have only nitrogenous base and sugar, that will tell us about the nucleoside. Okay. Usually in regular nucleotide, what happens? Uh, this is the nucleotide, whereas in RNA molecule, at the second prime carbon atom, hydroxyl will be there, and 3 prime hydrox, 3 prime carbon atom, hydroxyl will be there. That time we can say ribonucleotides. When are we going to say deoxyribonucleotides? When at the second prime carbon atom, when there is no hydroxyl group, that time we can say that is the DNA, deoxynitrogen, right? Triphosphates. So whereas this is the nucleotide, this is the nucleotide of DNA molecule where at the second prime carbon atom deoxy. Okay, this kind of nucleotides will be used in the DNA molecule. Okay, so deoxynucleotides are present in DNA, which acts as a substrate for various DNA polymerase, right? Enzyme which is going to add the individual nucleotides according to the template. So this is the normal DNTP. Okay, we can say this as normal DNTP. Okay, whereas in this Sanger sequencing, the major principle what we are using here is that is the dideoxy. Okay, here we are using the modified nucleotide. Okay, what are those modified nucleotide? That is nothing but the dideoxynucleotide. Okay, so deoxy, it means at the second prime carbon atom, we have removed the oxygen group, and third prime carbon atom also, Sanger has removed the oxygen group. So we can say deoxy. Together we can say dideoxynucleotide. So in short, in this is the DNTP. This is the DNTP. No, this will say which has normal nucleotide. Whereas in the Sanger's method, we are using modified. We are going to say this as DDNTP. D stands for di. Two oxygen molecules were removed from the second prime carbon atom and 3 prime carbon atom. What we have removed? Oxygen molecules. So dideoxynitrogen triphosphate is the principle behind in Sanger's method of DNA sequencing. What happens usually in the DNA synthesis, right? This is the single standard DNA fragment. Just imagine where we have the ribose sugar, then phosphodiester bond, right? And nitrogenous base you have here. And opposite nucleotides always base pairs with the hydrogen bonds, right, between the nitrogenous bases, and adjacent nucleotides which are going to interact through phosphodiester bonds. Whenever the whenever individual nucleotides which are added by the DNA polymerase, that time ultimately at the end we are going to get the one as the 3 prime hydroxyl group. At this end we are going to get the 5 prime phosphate. Always this end you are going to get the 5 prime phosphate, and the other end will be 3 prime hydroxyl group. Okay, so one DNA polymerase adds the individual nucleotide after the formation of phosphodiester. Here 5 prime phosphate, other side you will be able to get 3 prime hydroxyl group. Okay, to understand this concept, kindly you must go through the basic structure of this one, right? So for binding of the any nucleotide, available space are only 3 prime hydroxyl group, which is going to form a phosphodiester bond, and this is the fifth carbon atom, that is 5 prime phosphate. Only these two positions are responsible for the formation of phosphodiester bond, that is 3 prime hydroxyl group and the 5 prime phosphate group. For example, at the 5 prime phosphate region, if phosphate group is missing, no phosphodiester bond. Then at the 3 prime hydroxyl group, if this oxygen group is missing, then there is no phosphodiester bond. Okay, you must understand this one. Whereas in this particular case, there is always normal nucleotides will be having the hydroxyl group. Okay, whenever the normal nucleotides will be there, when the individual nucleotides which are added to the uh, uh, adjacent nucleotides with the end of the hydroxyl group, the normal nucleotides goes and binds here, right, through the formation of phosphodiester bond. Once DNA polymerase adds this kind of individual nucleotide to the with the growing chain, what happens in the addition of the nucleotide by the DNA polymerase? The final step is a nucleophilic attack here. Okay, nucleophilic attack by the 3' hydroxyl group on this. In this case, what is happening here? Here we be able to see the 3' hydroxyl nucleophilic attack can be seen here on this activated phosphorus atom. Okay, can make this one end. This is the 3' hydroxyl group, and this is the 5' phosphate group. Okay, in the action of DNA, like the formation of phosphodiester bond, nucleophilic attack by the 3' hydroxyl group on this activated phosphorus atom, which acts as the acceptor. Okay, a phosphodiester bond is formed and AMP is released. Okay, that is adenosine monophosphate will be released. Okay, where this here during formation of phosphodiester bond, ATP will be utilized, where ATP it will get converted into AMP with release of 2 inorganic pyrophosphate. Okay, here in this case what happens here, the reaction is driven by the hydrolysis of the pyrophosphate released during the formation of enzyme adenylate complex. Okay, that time what happens here during the phosphodiester bond, two high energy phosphate bonds are spent in forming phosphodiester bond in the DNA backbone with ATP serving as energy source. Okay, and what happens then here, eventually phosphodiester bond formation occurs. Okay, after the nucleophilic attack by this 3' hydroxyl group. So this is the normal DNA synthesis. Whereas in this Sanger's method, what happens whenever DNA polymerase adds the modified nucleotide like DDNTP, okay, like DDNTP, when DDNTPs were added to this particular region, what happens? Termination will occur and no further reaction because they lack 3' hydroxyl region. Okay, if 3' hydroxyl group is absent, further no reaction. Though there is an incoming nucleotide which is added by the DNA polymerase enzyme, whenever enzyme add modified DDNTPs, there won't be further reaction or synthesis, then chain termination will occur. A chain will stop. That's why this method was called chain termination. Okay, as 3' hydroxyl group is absent, there won't be any further addition of new nucleotides in the polynucleotide chain. Okay, the reaction would finally stop. Okay, further there won't be any kind of reaction which is occurring there. Hope you understood what reason the reaction has stopped. Why? Because these dideoxynucleotides are used as triphosphates and can be incorporated in a growing chain, but they can terminate synthesis as they cannot form phosphodiester bond with the next incoming DDNTP. Okay. The same thing which has happened here, when DDNTP is added in a DNA synthesis reaction, DNA synthesis will terminate with the incorporation of DDNTP. Okay, we can use DDNTPs of maybe DDGTP, a DDCTP, a DDCTP, or DDTTP. It means in general we have mentioned here the as DDNTP. The same thing DDNTP can also mention. We have 4 nitrogenous bases. You can mention DDATP, individual modified adenosine nucleotide. Then we can also mention DDTTP, that is thymine triphosphate. Then we can also use in the reaction tube DD GTP, that is guanine triphosphate, and also we can use DDCTP, that is cytosine triphosphates. Even we have normal DNTPs, right? We can also write this as the normal DNTPs. Normal DNTPs like the same thing, that is DATP, that is a deoxy adenosine triphosphate. These are normal nucleotides. Deoxy means at the second prime carbon atom, oxygen group is removed. Whereas in modified one, at the 3 prime also removed. So with the word dideoxy. So whereas next one is the DDTTP, and the next one is DGTP, and next one is the DCTP. You must understand here, these are normal nucleotides and these are modified nucleotides. What I'm going to say here is that when DNA polymerase cannot differentiate this normal NTPs and modified NTPs. Whenever DNA polymerase adds normal NTPs, the reaction continues. Whenever this DNA polymerase enzyme which adds the modified nucleotides, what happens? Termination will occur. Why termination occur? Because these DDNTPs in there we have removed the oxygen group from the 3 prime region, which is very much essential for the formation of the phosphodiester bond. Okay, when DDNTP is added in a DNA synthesis reaction by the DNA polymerase 1, then what happens? DNA synthesis will terminate with the incorporation of DDNTP. Okay, DDNTPs are also known as chain terminating nucleotides. These are chain terminating nucleotides. When DNA polymerase enzymes adds all these individual normal nucleotides, then chain begins. Okay, Sanger sequencing is also known as chain termination method or dideoxy DNA sequencing. Hope you're able to understand here. The main principle here is that modified nucleotide. This is the modified nucleotide. Okay, we have the this particular modified nucleotide which is going to terminate the reaction in the reaction tube. Okay, now let us understand what is this primer. Usually you'll get this question for 1 mark. Primers are short oligonucleotides. Oligo means few. Oligonucleotides which are responsible for the initiation, okay, or synthesis of the strands by the DNA polymerase enzyme. We need here the DNA polymerase enzyme, right? And we need a template DNA, right? So here what we are using here, the DNA polymerase enzyme. Here kindly make it, this is DNA, okay? DNA polymerase we need. And what kind of DNA polymerase is this? DNA polymerase is DNA template dependent DNA polymerase. It means this DNA polymerase can synthesize on the DNA molecule. So we can say this as the DNA template DNA polymerase enzyme. For example, RNA polymerase, whereas the RNA polymerase, they need RNA molecule as their template DNA molecule. Okay, whereas reverse transcriptase enzyme, again they need the RNA reverse transcriptase enzyme which synthesize the what? Uh, Complementary DNA molecule, right? From the mRNA molecule, so the RNA molecule, so they need the RNA template. Now let us understand more about this DNA polymerase first, what we are using in this DNA sequencing. We are using in the particular experiment DNA polymerase, okay? DNA polymerase we are going to put in the reaction tube, okay? So what is this DNA polymerase? DNA polymerase, which is going to add the individual nucleotides according to the template DNA in the reaction tube. Okay. So all 4 normal nucleotides are present. Okay. In the reaction tube along with the DNA polymerase. Chain elongation proceeds normally until when this DNA polymerase adds the natural normal nucleotides. By chance, if this DNA polymerase, okay, inserts a dideoxy nucleotides instead of normal deoxynucleotides, chain terminates. Okay. Further, there won't be any kind of the chain elongation process as that adds dideoxynucleotide. Okay. So this DNA polymerase enzyme will synthesize the complementary strand and later termination will occur. Okay. Now let us go through the four reaction tubes. Just keep in your mind this DNA sequencing mainly based on four reaction tubes. As this is in vitro DNA determination, okay, in this in vitro determination we are using the four reaction tubes. In all the reaction tubes we are going to provide the template DNA, then we are going to provide the primers, then we are going to provide the polymerase enzyme, then we are going to provide the normal DNTPs, okay, several normal DNTPs. These are the normal DNTP, and we are also providing the DDNTPs. Okay, DDNTPs are the modified nucleotides. Okay, let us go through this reaction of DNA sequencing. We are using the four reaction tubes here, each tube with a single stranded DNA. Okay, then further we are going to take the DNTPs, okay, normal DNTPs, then we need primer, then DNA polymerase and the reaction buffer. Okay, and also we are using here the DDNTPs. DD stands for dideoxy nitrogen triphosphate, which is one of the modified nucleotides which are radio labeled, because we need to understand the position of the DDNTPs. So these DDNTPs are radio labeled, which are incorporated into the growing polypeptide strand, which brings termination and cannot form phosphodiester bond with the next incoming nucleotides. Okay, even we can also label this primer, and also we can label DDNTPs. Just imagine these particular primers are labeled, okay, with the different colors, radioisotopes we can use. We can label primers, and we're also labeling these DDNTPs with the different radioisotopes which emits the different colors. We have four nitrogenous bases which are the building blocks of DNA molecule, that is adenine, guanine, cytosine and thymine. All these four nitrogenous bases were given different color in the DDNTPs, whereas normal NTPs are not radio labeled. Okay. After the process in the reaction tube, we are going to get the series of different length of DNA fragments in each reaction tube. Okay. Now let us understand this experiment here with these four reaction tubes of Sanger's method. Okay, here you could see this Sanger sequencing for reaction tubes. These are the common reaction components which are available in this particular four reaction tubes. Okay, so here in Sanger sequencing, we are making common reaction components like DNA template strand. Okay, we are going to provide the many copies of the DNA template strand. Okay, that is like this, the single stranded DNA we need. If we don't have a single stranded DNA, we need to denature by heating. Then in the reaction tube, we need to provide primer, many copies, and these DNA primers are also radio labeled. So this is the primer. Then we need the DNA polymerase enzyme. You need to provide the DNA polymerase enzyme. Then we need to provide the normal DNTPs, that is the deoxynitrogen triphosphates, which are in a large amounts. Individual nucleotides we need to provide. Okay, this is the common reaction components. In all these reaction tubes, you are going to provide the DNA template, DNA primer, DNA polymerase, and all these DNTPs. All this will be common reaction components in the four reaction tubes. Then what difference can be seen in the all the four reaction tube is that further in all these four reaction tubes, further you are going to add the, which is uncommon in this first reaction tube, you are going to add the only dideoxy ATP. In this reaction tube, only dideoxy ATP will be there, whereas in this reaction tube, only DDCTP will be there. In this reaction tube, only DDGTP. In this reaction tube, only DDTTP will be there. Just imagine in this particular reaction tube, okay, this is the single stranded DNA molecule here. Okay, here this is the single stranded DNA molecule, and here is the primer, and in this case DNA polymerase enzyme will be there. We've added here the modified DNA nucleotide DD-ATP. Adenine always base pairs with thymine, right? Cytosine always base pairs with the guanine. When we add here the along with normal DNTP is DDATP. This is dideoxy ATP. Whenever in the template DNA, if there is a presence of T nitrogenous base, this DNA polymerase adds the DDATP. It means for thymine, complement nitrogenous base is here, that is ATP. Whenever this reaction tube DNA polymerase adds DDATP, that time what happens? Further reaction will not happen. Chain termination will occur because in this dideoxy ATP we have removed the 3' hydroxyl group. Further, because 3' hydroxyl group is very much important for the phosphodiester bond. When there is no 3' hydroxyl group, further reaction will not begin or further reaction will not continue. Okay, so termination will occur. The same thing here also. Just imagine this is the template DNA molecule. This is the primer DNA which goes and attach, then DNA polymerase goes and binds there, right? In this particular template DNA molecule, if there is a presence of guanine nitrogenous base here, when DNA polymerase adds dideoxy CTP, okay, dideoxy CTP, further reaction will not be synthesized by the DNA polymerase because we have removed the hydroxyl group from the 3' region of this cytosine triphosphate. Then chain termination will occur. It means in this reaction tube, in the template, wherever the guanine will be there, their reaction will be terminated or reaction will stop. Same in this case also. Okay, this is template DNA and this is the primer. When DNA polymerase goes and attach, when they add the normal nucleotides, they will move whenever they find in the complementary nitrogenous bases. If there is a presence of cytosine, when the DNA polymerase adds the dideoxy GTP, further reaction will not continue. But when the DNA polymerase adds the normal nucleotide, reaction will begin. Or synthesis will be continued. Same thing here also. After the attachment of the primers here, DNA polymerase goes and binds there. Okay. When DNA polymerase adds the individual nucleotides according to template DNA, then reaction will continue. Whenever the DNA polymerase find here the adenine nitrogenous base here, when this enzyme which adds the DDTTP, DD means what? Dideoxy, oxygen removed, that kind of modified nitrogenous base when added, further no reaction. Okay, chain termination will occur. This chain will not be synthesized by the DNA polymerase enzyme. Okay, hope you understanding here. Let us understand through this reaction. This is the template DNA strand, and this is the primer, and this is the DNA polymerase. DNA polymerase adds the individual nucleotides, okay, according to the template DNA. Whenever this enzyme adds the DDNTPs, that is one of the 4 DDNTPs, that may be DDGTP, DDCTP, or DDGTP or DDTTP. When they add that kind of dideoxy nitrogen triphosphate, that time the reaction will terminate and enzyme will detach from that region and no further reaction. Okay, and this is the stopping of the DNA synthesis. These are called partially replicated fragment. Okay, so the same thing you can see here in the reaction 1 tube. This is DD-ATP. Wherever you find the thymine nitrogenous bases, whenever this DNA polymerase adds the normal nucleotides, they will keep on synthesizing. Wherever this DNA polymerase adds the this DD NTPs like DD ATP here, then reaction will stop. Okay. Then the same thing here in the reaction tube. Second, this is the template DNA molecule. In the reaction tube, the DNA polymerase added the C here, DDC here. Reaction has stopped. When the DNA polymerase add the normal nucleotide, okay, then there is no chain termination. Okay, whenever they add the this modified radio label nucleotides, only that time termination will be occurring. Okay, only during that time termination can be seen. In the same thing, you can also see that in the reaction tube 3. Okay, wherever the this particular DNA polymerase adds the modified nucleotides, their reaction has stopped. Okay, here also same thing has happened in the fourth reaction tube also. Wherever the DNA polymerase added the modified nucleotides, there what happened? Termination has occurred. Okay. In the sequencing process of terminating a single stranded DNA fragments to be sequenced is allowed to bind with a short radio labeled complementary fragment called a primer in the beginning. Okay. Following this, a primer bond fragments is then uh, what happens then DNA polymers keep on adding the individual nucleotides according to the template DNA molecule. Okay, then when this DNA polymerase adds the individual radio labeled dideoxynucleotide, that time reaction will stop. That is the basic principle behind this experiments. Okay, so you'll be able to determine like this. In the reaction tube 1, the N termination has occurred at the A region. Okay, whereas here after the addition of the A, okay, the termination can be seen like this, and here also you can see the termination can be seen like this. After these four reaction tubes, further this reaction tubes will be further subjected for the polyacrylamide gel electrophoresis. So next, once the for reaction tube incubation time is over, then what we are going to do? We are going to do the separation of components of each reaction tubes based on length of DNA fragments or size is determined through gel electrophoresis. Okay, the gel electrophoresis will be done on a high resolution polyacrylamide gel that where we can differentiate the strands that differ in length from single nucleotide can be determined. Okay, this can be carried out in a very thin polyacrylamide gel that is less than zero point 0.5 mm thick gel where during the process the double stranded DNA molecule that will be denatured by using the urea. Urea which denatures the new strands, okay, which is carried out at a very high voltage. In the beginning, the double stranded DNA, whatever newly synthesized DNA will be there, no? When the gel is heats up to 60 degrees Celsius, that time what happens? The double stranded DNA molecule will be denatured, and this high temperature which also avoids reassociation of strands. Okay, this is how once the reaction tubes for incubation is done, that reaction tube incubation content will be poured to this particular reaction well. The first reaction tube will be always in this well, second reaction tube in this well, then third reaction tube and the fourth reaction tube. After that, that will be run through electrophoresis methodology under the polyacrylamide gel electrophoresis. After the polyacrylamide gel electrophoresis, based on their size, based on their molecular weight, they'll be run in this electrophoresis. Larger fragments will be very near to the well, and smaller fragments will be very far to the from the well, or maybe are very near to this particular bottom region of the gel. Okay, so here you could see after running the gel, we can determine the bands. Okay, can be in this particular manner. Okay, this is the top and this is the bottom. Here you could see the larger fragment. In electrophoresis, mainly DNA fragments which are running based on their molecular weight, right? So here this is the direction of the reading the sequence in this particular case. Okay, so this is the bottom one and this is the top one. Smaller DNA fragments are at the bottom and larger DNA fragments are at the top. So how to determine this one? It means after the running this particular gel, okay, we'll be able to see the bands after staining. Okay, so further this polyacrylamide gel electrophoresis will be subjected to the autoradiography. Okay, where you could see here this is the smallest DNA fragment. Okay, this is the one by, okay, you can make good. The last smallest one is this one. So we can mention this as the A. Okay, always the smallest DNA fragment will move very fast because they have a less molecular weight. And the next one will be this one. Okay, after A. Okay, how we can determine C? Here in this case, we have put only the A nitrogenous bases of reaction tube, C terminated reaction tubes, G terminated reaction tubes, and T terminated reaction tubes. So wherever this fragment, wherever this line indicates, this particular line in this lane which indicates the presence of the adenine nitrogenous base, okay, then this line which indicates the presence of T nitrogenous base, and they have terminated over there. And next comes the here, that is G, and next comes here, this is the C, and next comes here, this is the A. Like this we'll be able to determine the arrangement of the nitrogenous bases. Okay, so same thing can make out here. First we found A, and then we found T, then we found G here. Okay, then we found C here. Like that you are going to determine the DNA molecule, and you are able to find, okay, this kind of after autoradiography. We have, because we have radio labeled the individual DD and TPs, and we have also radio labeled the primers. Okay, so whereas in this case, based on their fragments, okay, we are able to differentiate this, determine this particular nitrogenous bases according to the this particular um, gels which are on the electrophoresis. Okay, each uh, band contains some amount of DNA molecule in the gel electrophoresis. Okay. After gel electrophoresis, we are subjecting to the autoradiography to visualize the DNA bands of four lanes. That is, fluorescent dyes can be used, or UV trans illuminator we are going to expose, or we can also use the gel dog. Okay. Here we are going to compare the four lanes of bands on a gel to determine the sequence of nucleotide. Finally, technologists determine the sequence of nucleotide of unknown DNA fragment. Okay, nowadays even we automated DNA sequencing by using fluorescent dye is also available. So this is the DNA sequence what we determine through gel electrophoresis techniques after the auto radiography, and this is the original template stand what we used in the beginning. So template stands can be seen here, almost it's the same. Okay, this is how we have determined the uh, arrangement of the nitrogenous bases in the DNA molecule. For example, if this is the band, in this band we are going to get such type of DNA which is a very small DNA molecule or low molecular weight. As and when you see here from the bottom to top, there is an increase in their molecular weight. Based on the increase in their molecular weight, they are going to settle in that particular lane of that particular gel, for how we have, however we have run. Okay, this is the overall reaction process. Okay, in the Sanger's method of dideoxy nucleotide method. Okay, first we here in the beginning we have taken here the double stranded DNA molecule. Okay, once we take the double stranded DNA molecule, we can go through denaturation. Okay, we can go through the denaturing the DNA or gene inserted into an M13 vector to get single stranded DNA molecule. We can expose to the M13 vector, or we can also treat NaOH to denature the DNA molecule, or else we can also go through the heat denaturation process, because whenever we have a double stranded DNA, in double stranded DNA, any DNA polymerase cannot go and sit and cannot synthesize the DNA molecule, right? Compulsory DNA polymerase need single stranded DNA template, and they need the double stranded initiation region in the beginning. They need, then only DNA polymerase goes and binds. That's why we need to denature the DNA molecule for the annealing of the primer, right? Once we denature the DNA molecule, you are going to get this kind of single stranded DNA molecule. Okay, one single stranded DNA molecule is ready. You are going to say this as template DNA. Okay, so in the reaction tube, you are going to provide radio labeled primers, Primers are required for the detection of the newly synthesized DNA after electrophoresis during autoradiography. Okay, so primers are required. Primers can also be radio labeled. Then we require here DNA polymerase, which play a very important role for the synthesis of the DNA molecule. So in the reaction tube, we are going also providing the DNTPs, that may be a DATP, DGTP, DCTP, DTTP. Okay, during the synthesis process, DNA polymerase need all these nucleotides which acts as a substrate. Additional, other than all this, to stop the reaction, okay, in DNA sequencing method, that too in Sanger's method, we are using the modified DNTPs, okay, that is nothing but dideoxy adenosine triphosphate. I've already given this particular structure. Dideoxy means at the 3' region, we have removed the hydroxyl group. When we have removed the hydroxyl group from there, further no phosphodiester bond, okay. Same kind we are providing here DDGTP, DDCTP, DDTTP. All these DNTPs which are colored in give different colors. This may be a green isotope, this may be a red isotope, it may be a pink isotopes, and this may be a yellow isotopes. Okay, so all these components you are going to add to the 4 reaction tubes. So whereas this radio labeled primers, DNA polymerase and DNTPs in a reaction buffer, up to here this will be common. Okay, in all the reaction tubes you'll be able to see single standard DNA template, DNTPs can be seen, primers can be seen, and DNA polymerase can be seen. Okay, only exceptional case is that in this first reaction tube we are adding only one type of DDATP, and here one type of DDGTP, and here one type of DDCTP, and here one type of DD TTP. Okay, it means in this reaction tube, wherever reaction occurs, no, that will be after the addition of DDATP. Here at the after adding DDGTP, DDCTP and DDTTP. Okay, so I've written here termination at DDATP, termination at DDGTP, termination at DDCTP, termination at DDTTPs. Okay, so we can also write here DDNTP stop reaction or brings chain termination. Okay, so this is the template DNA strand. To this template DNA strand, primers attachment will be happening. I've given very lesser arrangement of the nitrogenous bases, less small DNA fragment, but usually we need the larger DNA fragments because primer attachment also will be happening. To make you understand, I have given the small DNA fragment here. So just think here ATGC, ATGC is their sequence. Okay, in this reaction tube, this DDATP will be added whenever they find thymine nitrogenous base. Okay, to this thymine, they'll add the dideoxy ATP, then reaction stops here. DDGTP, they terminate the reaction when they find the cytosine. DDCTP, they terminate the reaction when they find the G, right? DDTTP, they terminate the reaction when they find the adenosine or adenine nitrogenous base. Okay. So this is 5 prime. Okay. Wherever you can see this is the template DNA strand. And when DNA polymerase adds the thymine here, there is no reaction. When this DNA in this reaction tube, when this DNA polymerase adds the radio labeled modified DDATP, then reaction terminated in this reaction tube. When they terminate here, see their length of the DNA fragment is very less. Okay. Next, this is the DNA. It means in the reaction tube, many such kind of template DNA strand, primer, and synthesizing template DNA strand, primer, and DNA synthesizing. Many can be observed here. We are not providing only a single, 2 or 3 template strand. As I told, several DNA fragments will be there. Okay. So here you could see termination can be seen here, whereas in the reaction tube 2, we can see here, right? That is the DDGTP, whereas this the DDGTP termination has occurred at the when there is an addition of DDGTP, termination has occurred here, and there is an addition of DDGTP here, termination has occurred here. See the length of the DNA fragment. This length is quite bigger than this one. According to their length of the DNA fragment, they'll settle in the gel electrophoresis. Okay, whereas here this length of the DNA fragment is very larger, so this will settle very near to the well. Okay, whereas in this case termination of DDCTP, here you could see at the this particular region to the guanine nitrogenous base, and here also whenever the guanine they find, there's a termination has occurred. But here also guanine is there. When there is the addition of the normal nucleotide, there won't be any kind of a chain termination. When the DNA polymerase adds by chance this modified nucleotide, then termination will occur. Okay, see that see this length of the DNA fragment. This is quite different, and this is different. But all the template are in the same size. Okay, all these and temperature are the same size, and in this case also DDTTP, this is the template DNA. Whenever the DNA polymerase adds this modified nucleotide, then termination of the reaction occurs. This with only differ with only one nucleotide. Okay, and the next one is this is the DNA template, and this is the addition of the complementary nitrogenous bases. See the length of the DNA fragment. Okay, once all this incubation of the four reaction tubes done, no, further we'll be going through the electrophoresis techniques. So in this electrophoresis techniques, in the reaction mixture, we are separating this particular DNA fragment. Okay, so in this case, what are we doing here is that in this gel, we are separating the DNA fragments based on their length or molecular weight of the DNA fragment. Okay, so here we are using the gel of less than 0.5 mm. We are using this particular gel here. Okay, all the reaction tubes by taking the micro pipette that will be loaded to this particular reaction well. First reaction tube will be loaded in this well. Second reaction tube will be loaded here, and third reaction tube will be loaded here, and fourth reaction tube will be loaded here. After loading, we are going to perform the electrophoresis. Okay, that is we are going to perform the gel electrophoresis. Okay, in the gel electrophoresis, what are we doing here? We are in this case, what happens? Migration of the charged molecules under the influence of electric field, which moves from negative charge to the positive charge. Okay, from negative to positive we are using. Why we are negative connected to the wells? Because as our DNA is negatively charged, they move from negative to positively charged condition. Okay, here the gel contains the urea, which denature the DNA molecule. So this is the DNA molecule already that they are in the double standard condition. Okay, so here when we denature, what happens? All the template strands will be very near to the well, which are of the same length. Okay, and here after the running of the gels, we can see the bands in the gel lane, okay, which indicates the position of terminated DNTPs. After running the gel, okay, we can see the bands on the DH particular gel. The presence of the bands in that particular lane of the well, that indicates the presence of the that particular DNA. Okay, through that we are going to determine the nitrogenous bases. For example, here the very fast or very low molecular weight DNA molecule which move faster. Okay, and that will be very near to the bottom because they have very less fragment of DNA. For example, here you could see one band, here you can see one band, and here you can see one band, and here you have one band, here you got one band, and here you got, and here you got. So now we are going to determine the alignment of the nitrogenous bases, which is the very far less molecular weight DNA band here. This is the one because which is very less molecular weight. Based on their size of molecular weight, we are differentiating the DNA molecules in this electrophoresis. Okay, so this is the DNA molecule. Okay, that is T. T means this one. Okay, okay, I told here, no, in electrophoresis, DNA sequencing, which could differentiate or determine the DNA fragments which differ in single nucleotide. So you could see here, this is the first band in this gel electrophoresis. So we are going to write it here, this as the T. This is the T well. Okay. So this is the T well. Right, this is the T well, and this is the C well, CTTP or CTP. This is the GTP well, and this is the DDATP well. So here we can determine that the very small fragment is the T, and the next fragment is here the, this is A. So this is A, okay, and the next one is C, the next one is G, okay, and the next one is T, and the next one is here A, and the next one is C, and the next one is G. Okay, this is the determined order of nitrogenous bases, okay, that we can compare with the this original DNA fragments. So we got here the complementary nitrogenous bases. Through this DNA sequencing or Sanger's Dideoxy method of DNA sequencing, we have determined the unknown sequence of the DNA molecule. Okay, you can also keep this in your mind. The same I have written here in the pen, and here I've used the color pen. Okay, you can go through this or that one, or you can also go through this particular tabular column for the DNA sequencing enzymatic method. Okay, and this is how wherever termination has occurred, there we are getting the different length of the DNA fragment. This different length of the DNA fragments can be observed through the electrophoresis. Okay. So what are the limitations here? Sequence only for 900 nucleotides or base pairs at a time. Nowadays, maybe you may find the advanced technique also. And this particular DNA sequencing is time consuming and laborious. Okay. Nowadays, Sanger's automated DNA sequencing fluorescent molecules will be available, which are used, and whereas in this case, in this Sanger's automated DNA sequencing, instead of 4 reaction tubes, they are using the a single tube to separate those different length of terminated DNA fragments can be done. Okay, this can be detected by fluorescent detector. And in this case, nucleotides can be labeled with enzymes, okay, or maybe antigenic substances, or radioisotopes, or chemiluminescent moieties can also be used, or fluorescent dyes can also be used that bind with high specificity. Okay, to conclude here, Sanger's DNA sequencing requires template DNA, okay, that is pure and that should be homogeneous. The DNA is made single stranded by cloning with phage or M13 or by denaturation process. Okay, DNA polymerases copy the single stranded template, initiating the synthesis at a point which are bound by oligonucleotide primer. Okay, 4 sequencing reaction tubes are prepared, each containing a different DD NTP and different DNTPs and DNA polymerase in the reaction buffer. Okay, DNA synthesis initiated from the primer annealing and is terminated at random incorporation of DDNTP. Okay, each reaction generates a series of DNA molecules of different length. Okay, different length terminating at positions corresponding to the presence of one of the base. One of the base means only DDATP. Whenever this particular DDATP will be added, there you can see the chain termination. Okay, each uh, reaction generates a series of DNA molecules of different length terminating at the positions corresponding to the presence of one of the base in the template. The DNA molecules are separated by size on polyacrylamide gel and visualized by autoradiography. Okay, after the autoradiography, you'll be able to see this kind of DNA ladder, and the sequence is read from the identity of the terminal base in successively longer fragments. From that, one can make a DNA sequence, or one can determine the DNA sequence. Okay. Hope you understood the DNA sequencing. If you feel like this is in detail, I'll be back with very short DNA sequencing information. Okay, this is Dr. Pratibha K signing out for today. Thank you for watching.